So thank you very much for the introduction, and thanks for having me. I'm a postdoc. I'm a computer scientist, and I have worked for a couple of years now on RNASeq. I'm going to tell you a little bit about what RNASeq is in case there's some non-next-gen sequencing folks in here, which I'm sure there are. And then I'm going to tell you about what I think is kind of the great opportunity and also the great challenge of RNASeq, which particularly relates to visualization and data exploration. I'm not a visualization guy. But in my experiments and in my analysis of RNA-seq data sets recently, I've come across some sort of obvious sticking points with interpreting this kind of data. And I think that there's some, maybe not easy to solve, but at least straightforward problems to state that come up during isoform resolution RNA-seq. So RNA-seq is an assay that, at least in theory, can tell us the abundance of just about every transcript in the transcriptome for a particular RNA sample. And that's a lot of power. Naturally, we want to know, OK, what are the limits? What can we do with this information? What do we want to do with this information? So you just heard about some gene discovery and transcript discovery applications of RNA-seq, which are very exciting. I'm not going to talk about that at all. I'm only going to talk about expression analysis right now. And I should also say at the beginning that I work in mostly human and sometimes mouse. And so I'm almost always going to assume that the transcriptomes we're talking about are alternatively spliced, and each gene has many isoforms and things like that. OK, so differential gene expression is kind of a bread and butter analysis for just about everybody these days, especially on a small scale and even on a large scale with microarrays and things like that. But only recently has it been possible to measure changes at the isoform level, which can tell us something about how, not only how the genes are sort of being regulated up and down between conditions at a whole gene level, but also how individual isoforms of those genes are being regulated. So here's an example of the kind of regulation, kind of transcriptional regulation phenomena that we might be interested in. So suppose you've got a gene that's got two different promoters, and you're interested in figuring out how a certain set of transcription factors are regulating the behavior of a particular developmental system or something like that. And you see that this very important gene actually has two promoters, and the transcription factors that you're studying kind of fall into two different cohorts, one of which binds at one promoter and one of which binds at the other promoter. And it looks like when you look at your RNA-seq data or your expression data that there's actually some sort of a promoter switch, okay, so that the gene is preferring one promoter in one condition and the other promoter in the other condition. This is a super interesting sort of example because this is an opportunity to relate isoform level transcriptional regulation back to all of the proteins and all of the regulatory elements that would be affecting this change. So these kind of things are very exciting and possible with RNA-seq because we can measure the abundance of every transcript. Another sort of classic area of interest, right, is splicing. So suppose we've got a gene that has 3 exons here, and if I do this, I get the blink. So we've got exon 2 here, which is actually a cassette. It's an alternative exon, and there's another, say in another condition, this gene actually, when the splicing machinery is coming along and processing the primary transcript, the exon along with the 2 flanking introns get excised as one unit. And if this is part of the coding sequence, this change is probably going to have some very obvious and potentially very important functional impact on the proteins that are coming out of this gene. And I'll show you some examples at the end of the talk where we think this is probably the case. OK, so as Kai mentioned, I have worked on tools for sort of next-gen sequencing analysis as a whole, but particularly for RNA-seq. And all of this stuff is sort of open source. So if you're new to RNA-seq or if you're thinking about getting into it, I recommend that you take a look at these tools because they're widely developed or sort of very actively developed by a large team of people. We spend a lot of time trying to get this stuff right, and this is sort of a growing collaboration. Most of these people are actually still very involved in these projects. So I encourage you to take a look at these tools if you're thinking about doing an RNA-seq experiment. So the basic kind of, now I just want to get into sort of what RNA-seq is and how it works, how it measures abundances for each transcript in the transcriptome. So the fundamental principle of RNA-seq is that you take your RNA and you turn it into a sequencing library and you put it into the sequencing machine, and what you get back are reads, sequencing reads, millions and millions and millions of them usually. And the basic principle is that the more abundant a particular transcript species is in your input RNA, the more reads you're going to get back from it. So the name of the game when we're doing the analysis is going to be to sort of figure out which read came from which transcript and count them all up and then use that somehow to infer what the expression levels were. There's a little bit of a wrinkle, though, which is that because of the way most RNA-seq protocols work, you'll also get more reads from longer transcripts. So 2 transcripts present at the same abundance, you'll get more reads from the longer one. So we have to account for that fact when we're converting these counts into expression levels. OK, so that's kind of wrinkle number 1. Wrinkle number 2 is that, especially when we're talking about alternative splicing and multi-isoform genes, we often don't know which read came from which transcript. We don't know, if we're looking at a read, we can't always tell which transcript it came from. So, for example, the reads here in black could have come from any one of these 3 isoforms of this cartoon gene here. We can't tell just from looking at how they align which one's the right one. Sometimes we can. This yellow spliced fragment here actually spans a splice junction that's a distinguishing feature of the gene. So we know in that case, but often we don't. And that's going to be a big problem. We're not going to be able to just count things up. If we want to measure the abundance of every transcript, we're going to have to kind of guess how many reads came from each isoform. OK. So cufflinks is a program that does a couple of different things. It can discover new genes and assemble reads into full-length transcripts and tell you about new isoforms and that sort of thing. But it also can tell you, it also can sort of solve this abundance estimation problem where you give it your reads and your transcripts, and it tells you its best guess for how abundant each of those transcripts are. And the way it works, just at sort of 10,000 feet, is that inside the program and inside the algorithm is a mathematical description of how RNA-seq works, the actual sequencing process. It's very simple, makes a lot of idealized assumptions, but it works quite well. What it does is that it takes that model, and that model comes with a little function, a function called a likelihood function. That function takes as input transcripts, reads, and your proposal, or the program's proposal, for how abundant those transcripts are. And it returns a number. And the number is how likely you would be to get the actual reads you got in your experiment, given those transcripts and your proposal for the abundances. So what the program then does, is the algorithm then does, is it takes that model and it searches the space of all possible assignments of abundances to transcripts and looks for the abundance assignment to all the transcripts that maximize the likelihood of getting the reads you actually got in your real experiment. So what it's producing is a best guess, in the statistical sense, a best guess for how abundant each transcript is. And we're going to see in a second why this is important. Not only does it give you the best guess, but it also gives you its own assessment for how confident it is in that guess. So you can imagine that if your gene is being sequenced and it only has 5 reads coming from it, has 10 isoforms, the program's not going to be terribly confident about its best guess. But if you have 5 million reads, it might be quite confident. And as we'll see when we're analyzing different conditions, that's going to be a critical issue. OK, well, all that sounded very complicated. I just described the statistical model, and I told you that there's this sort of counting operation. So maybe we can just punt on that, on the isoform stuff, and just work at the gene level. So actually, a lot of very popular methods for doing differential expression analysis with RNA-seq, what they do is they just kind of count up the reads on each gene in each condition, and then directly compare the change in the counts. And most of the time, this works just fine. But if you have substantial isoform switching or anything like that, like I showed at the beginning, you'll get a very wrong answer for even how much the gene is changing. Forget the isoforms, but even how much the gene is changing. I'm not going to take you through all these examples, but I will say that just by sort of moving the reads around on the isoforms and comparing the simple method to kind of the cufflinks method, which would be to estimate the individual isoform expression levels, add them up, take that as the gene expression level in one condition, do the same thing for the other condition and compare them, you can actually get drastically discrepant, qualitatively different answers for even the direction of the fold change. So depending on how the example works, you can see that the simple counting method sort of says the gene's going up when in fact it's going down. And so that's sort of the consequence of that, I argue at least, is that even if we just want to do differential gene expression, we need to be working at the isoform level first if we want to make sure we're getting the right answers. So, all right, now let's talk about sort of the basic problem of comparing 2 samples. I've sort of outlined how to do the analysis at isoform resolution for one condition. If we have multiple conditions, what we're going to do is we're going to run the program. We're going to figure out how many reads probably came from each transcript in each condition. And then we're going to convert those into expression levels and compare them. And in this case, we say, OK, well, we've got more reads that came from this particular transcript in condition A. So we see this, we observe this change. And now the question is that change statistically significant? Because this sequencing process is a random process. The sequencing machinery is random in some sense. It's producing random numbers of reads from each library. And also, of course, these conditions have some biological variability. If we were to collect multiple replicates for A and multiple replicates for B, there'd be some variation between them. And that variation just within the condition might exceed the change that we see in the reads. So we need some way of describing how much variability we can expect. And then use that description to assess p-values and such on any changes we see. So until recently, there was no real program that would kind of deal with both sources of variability that come up when you're doing isoform resolution analysis. Most of the work had been done that focused on sort of modeling how much intra-replicate variability, intra-condition variability you get, and sort of been ignoring the problem of this mapping uncertainty, where when you're sort of guessing at how abundant the isoforms are, you don't always know because you have this statistical inference procedure going on. So CuffDiff2, which is a new algorithm that we just sort of published a couple of months ago, combines both, sort of addresses both of these statistical sources of noise or variability under one model and one hood, so that you can actually, analyze your data at isoform resolution in a statistically principled way. Now that we can do that, now that we can do isoform resolution calculations and also assess significance of changes, we can do neat stuff like look for the kind of regulation that I mentioned at the beginning. So suppose we've got this idealized gene where you've got two transcripts, a blue and a yellow, that come from the same transcription start site. So these guys are they're being processed from the same primary transcript, and the splicing machinery is just doing something differently with them. If we look at not just their absolute expression levels and changes in those levels, if we actually look at the levels relative to one another, we might see, for example, that in condition A, the blue and yellow isoforms are about equal, but in condition B, there's much more of the A isoform. And that's an example of differential splicing. So by taking kind of biologically meaningful groupings of individual transcripts, we can make some inference about what's going on at sort of a post-transcriptional or a transcriptional level. I'm glad that Paul already showed pie charts because I was embarrassed about this. So the other sort of really substantial problem with RNA-seq and with analyzing RNA-seq data is that, first of all, these experiments produce an absolute just ton of data, millions and millions of reads. You have typically many conditions. And then now we're talking about isoform resolution. So you've got 100 or 200,000 different splice variants of your 25,000 different genes or 50,000 genes. If you're interested in link RNAs, that's an extra 10,000 genes. The scale of the analysis is a little bit overwhelming. And mining the data, interpreting the data, even making figures out of it can be onerous. So my buddy Loyal Goff, who's another postdoc in the RIN lab, actually decided to tackle this problem by building a new package called Cummerbund, which is the newest of the Tuxedo tools. And Cummerbund is a program that its main mission in life is to help you look at your favorite gene in as easy a way as possible, but in a way that is scriptable and reproducible. So what Cummerbund does is it takes all of the complicated output from CuffDiff, which is 15 different tab-delimited text files, and boils them all down into a single SQLite database that's lightweight enough to fit on your laptop. And then, so that way you don't have to be working on a server or anything like that. And it lets you quickly make almost publication-ready plots for your favorite genes at gene level, at isoform level. You can look at, you can do sort of classic visualizations like heat maps as well as some sort of more advanced stuff. So this is kind of what our whole, the whole RIN lab has kind of moved over to this thing. Rather than interacting with their CuffDiff output directly, they're doing everything through Cummerbund because it just makes life a lot easier, especially for people who don't have sort of bioinformatics training. So here you can see I'm showing an example. Just with one little line of this kind of script language, you can produce this plot of tropomyosin 1 and its activity during a time course of muscle cell differentiation. Tropomyosin 1 is well known to have a skeletal muscle-specific isoform, which is coming up after 60 hours really clearly. So that, just right away, you can kind of look at your favorite genes very easily. So now in the remaining time, I hope I have some remaining time, I'm going to show you a few examples of interesting things that we found from recent RNA-seq analyses, just to give you an idea of the kind of regulation that we're looking for, and in particular to highlight what I think is the big open problem. So when I first got to the lab, I started working with David Hendrickson, who's a postdoc in the RIN lab, who works on Hox biology. And he was looking at Hox genes in adult cells. Hox genes, of course, are extremely important developmental transcription factors. They're responsible for body patterning and that sort of thing. Not too much known about how they work or if they work in adult cells. So he knocked HOXA1 down, for example, in lung fibroblasts and found, very surprisingly, that within 72 hours, even after 48 hours, just about all the cells were dying and on their way out, and very few remained. And he was able to reproduce the same result in HeLa. And after very carefully checking for off-target effects and things like that, he did some extremely deep RNA-seq and microarrays to boot to compare. And we kind of rolled the whole experiment into a paper that talks about cuff diff. As we were exploring the data set, it became pretty clear that the cells that were remaining on the plate after HOXA1 knockdown were kind of stuck in G1. They were stuck in a particular phase of the cell cycle. And all the other sort of cycle phase specific gene sets were greatly depleted. So we started looking at classic regulators of the cell cycle and cell cycle associated genes. This is CDK2. And we noticed, of course, that CDK2 is upregulated in response to HOXA1 knockdown, which is consistent with a G1 block. And just about all of that increase is attributable to this blue isoform here, which differs from the other two in a very interesting way. It includes this cassette exon, which itself contains the lion's share of a critical protein-level feature called the activation loop, which is required for CDK function. So you can see that these isoforms are being independently regulated, and they're being regulated in a way that would have pretty obvious functional impact in terms of the proteins that were coming out. We looked at other sort of cell cycle genes and found similar phenomena. So TBX3, which is a transcription factor that's related to, that sort of has roles in apoptosis and other things. We could see that it's differentially spliced, and the splicing event affects the T-box DNA binding domain, so its ability to bind DNA may be somehow different. And there are other examples from this experiment. So switching to a different example now, this is something that wasn't done in the RIN lab. This was done in Alex Meisner's lab. Alex Meisner has been working with his two grad students, Casey Gifford and Michael Ziller, for several years now, developing a really cool system, an in vitro model of human ES cell differentiation. So by taking ES cells cultured in teaser and ES media and replacing that media with differentiation media with specific additives, they can actually send those ES cells down one of 3 different paths corresponding to the 3 different germ layers present in very early human development. And they noticed when they started looking for, of course, then they did RNA-seq on, on all 4 conditions and found a number of, of course, thousands of differentially expressed genes, but also hundreds of differentially spliced genes or genes undergoing promoter switching, and saw that, there's one isoform, which is the major isoform in ES cells, and that isoform is basically lost and replaced by another immediately following differentiation, which is interesting because the green isoform here contains 3 exons that the purple one lacks. And these two contain a large portion of the catalytic domain of DNMT3B. DNMT3B is a DNA methyltransferase. DNA methylation, of course, is extremely important for maintaining pluripotency, and DNMT3B is actually embryonic lethal in mice. So you got a loss of the catalytic domain upon differentiation, and also this purple isoform is believed not even to be able to bind DNA, so it requires some cofactors in order to bind. So again, like a major protein level change, which would have sort of clear functional import downstream. Ezgi, who's a grad student in our lab, works on the HNRP proteins, and she's been looking particularly at HNRPU. These are nuclear matrix proteins about which very little is known. They're believed to be involved in splicing, but really not in a super clear way. And so she knocked down HNRPU in HeLa cells and observed, I'm not showing it, but observed almost no change in expression in this gene called muscleblind2. But when she looked at the isoform level, she saw that even though the gene wasn't changing very much, the distribution among the isoforms was changing very dramatically. So she was seeing a complete loss of this blue isoform here, which includes the complete structure of muscleblind2 in response to loss of HNRPU. So if she hadn't been looking at isoform resolution, she would never have seen a change in this very important gene. I bring it up because there was actually a really nice paper recently in the last couple weeks, maybe a month or two ago, written by Eric Wang from Chris Burge's lab, focused almost entirely on muscleblind 1 and 2. Muscleblind 1 and 2 is a very important gene in myotonic dystrophy, and is also itself known to regulate splicing and RNA localization. So you've got a differential splicing event in a splicing regulator, which is known to have lots of important functions in various diseases. So, if you want to see an extremely careful and high-resolution analysis of RNA-seq data, I encourage you to take a look at this paper. It's just super cool. So the challenge that I think is kind of the main challenge that we face when we're looking at this type of regulation is that we need to incorporate splicing structure from one database, say UCSC, with protein-level feature annotation from other places like PDB or PFAM or NCBI directly. And then, of course, we need to overlay expression data at the isoform level into single figures and visualizations. And we have no good way of doing this right now. I mean, Cummerbund is great in the sense that it's fast and lets you plot your favorite genes, but it doesn't know about all this other data that's out there. And I think this is basically a data exploration and visualization challenge. I think we need better tools for combining all this information together, because without it, you can't really interpret these differential splicing and promoter switching events very easily, because there's lots of them, and you don't know which ones you want to focus on. So just to sum up, differential analysis at isoform resolution is possible and maybe even necessary, even if you want to do just gene-level stuff, but it's very hard. And that's because these sequencing experiments tend to involve tons of data and often complicated statistics, which need to be propagated to you, the user, so you can assess what's important and what's not. I didn't really talk about this, but it is an issue that often comes up for isoform resolution stuff. Our annotations of our transcriptomes are constantly in flux. GenCode, which is about the most comprehensive annotation that I know of, comes out with an update about every month or two. And they add lots of new transcripts and change IDs. And that makes it hard to connect results and findings across experiments over time. I think most seriously, there's relatively little isoform resolution functional annotation out there, and we have almost no good way of combining it together with our expression data yet. So I think this is the main challenge. I just want to acknowledge my lab mates and my mentors. So Dave Hendrickson and Laurel Goff contributed a lot to the software and the slides that you saw today. Others from the lab certainly did as well, such as Ezgi, I showed you some of her data. And John is a great supporter of all this isoform resolution RNA-seq stuff for sure. And the whole sort of CuffDiff project was done in collaboration with John's lab and also Leo Proctor, my advisor away from home at UC Berkeley. So thank you very much, and I hope I have a little bit of time for questions. Maybe I did.